Good afternoon, everyone. Wake up. I just had lunch. First one after lunch with some really exciting information brought to us here today on the 6PPD track. You've been with us from the beginning of today. You learned about the issue. You saw that wonderful video made by Whatcom County. Saw Curtis's presentation about solutions that have been found in the lab, in the field, and Amy's presentation. And now Dylan's going to talk about some field work that's been done to help us address 6PPD-Q in our urban and suburban environments. Here in front of me, I have Dylan Ahearn. Dr. Dylan Ahearn is a hydrologist who spent the past 20 years studying the environmental consequences of human alteration to aquatic systems. He is currently Director of Science with Herrera Environmental Consultants and an adjunct professor of geology at Edmonds College, and I might add, a board member of Stewardship Partners. So we'll plug in there for that. Dylan, go ahead and take us into this presentation. Thanks.
You can see that. As you can see from the names up there, I'm also presenting with Chelsea Mitchell, who's been highly featured in this conference for having not been here, but unfortunately she couldn't make it. She's also co-author with Curtis on his presentation. She's also featured in that nice video this morning. So I was happy to collaborate with Chelsea on this study here. You know, we've heard today a lot about how great bioretention is, and we've known this for a while, right? It's very good at treating things like fecal coliform, which is hard to treat with most BMPs. Great for TSS and for zinc, but also cool for like placemaking and habitat and traffic calming, all these wonderful things. So of course, our research is focused on bioretention a lot in this region. Turns out it's also really good for the removal of 6-PPD-quinone. We've seen that in a study presented by Curtis and by Amy as well. Really exciting that all these data are coming out now, and we're going to get into those data. But before that, I'm going to take a step back and talk about 6-PPD-Q not so fun facts. I don't think this slide has been shown yet in terms of like the background slides on 6-PPD. And this one is from Zhenyu Tian and Ecology, a paper they put together in 2022. And what it is it's linked chemicals and the organism that they are toxic for and their LC50s from the most toxic at the top on down. Does anyone know what most of these chemicals are on this list? Are they familiar, some of these names? That's tiny up there. Y'all can't see that at all, probably. Bunch of pesticides, right? What's not a pesticide is the number 2 most toxic thing on there, which is 6-PPD-quinone. That's just really bad luck for coho salmon, right? Designed as a plasticizer for tires. It's an oxidized version of that, just happens to be highly toxic for coho salmon. And just how toxic? Well, we're talking between 40 and 90 nanograms per liter is the LC50, depending on the life stage. Converting things to the universal units of Olympic-sized swimming pools, that is 40 drops in 20 Olympic-sized swimming pools kills half the salmon in there, half the coho. It's pretty tough. I have this slide too, and this was presented already at this conference, but I think it's an important one because when I talk about 6-PPD-Q in places like California, sometimes the question is, why do we care? We don't have coho down here, like the range is down to Santa Cruz roughly, and coastal, right? But research is coming out now that there are, not only acutely toxic effects at much higher concentrations, but sublethal effects for many more species that have range across the whole country, right? So when you see this, you saw the videos today, like when you see that happening with coho salmon, that's an indication that stuff's pretty bad for fish in general. And those data are being collected now, and like just how pervasive this issue is, what are the sublethal effects for which species. And I think what did we hear, that it actually, the coastal cutthroat almost as toxic as for coho. That was news to me today. So all this information is coming in, and we know it's a big problem. We should care about it no matter where we are in the country. Luckily, back to bioretention. Bioretention is pretty good at removing 6-PPD-Q. What it's particularly bad at is nutrients, right? And that is the standard bioretention soil media spec, 60% sand, 40% compost. We know compost leaches nutrients. That's why the plants love it, and that's a good thing. But we didn't know, the magnitude of this issue until the city of Redmond started looking at it pretty closely because they wanted to treat their stormwater. You know, this is 2013 now. Everyone wanted to do LID in their jurisdictions, but they drink their shallow groundwater supply. Steve knows all about this, and wanted to make sure that the water, you know, is filtered to a high degree before they infiltrated. But what they documented was with an underdrain in these systems, and I think it's up to 7 bioretention systems full scale in the field that they ended up monitoring, is that there's just a ton of nutrients coming out the underdrain. You know, this soil media spec, 60% sand, 40% compost. If you don't have an underdrain, it's awesome, right? Because you have dispersed nutrients going into groundwater. That's not a big deal. It won't be too big a loading in any one spot that you'd have to care about it. But when you have an underdrain, you're just routing that straight to a receiving water, right? It's a filter, and especially if it's lined, right? And as we started implementing bioretention across the region with our, you know, difficult soils, people started designing more and more fully lined bioretention systems with underdrains. And at that point, it's just a filter. And really, it's your last chance to get it right at that point. Like you've messed up 100 ways in your basin, you've put in the pavement, you brought in the cars, you brought in the pollution, you routed everything. And now you have 18 inches of soil between you and the stream. It's your last chance to get it right. And so this is what really spawned a bunch of research to get that 18 inches really, really right. You know, we should put some more engineering into that to fix that issue. When that was documented in Redmond, that amount of nutrient export, Ecology responded saying, you know, reminding everyone that if you're within a quarter mile of a sensitive receiving water, don't do bioretention. If you have an underdrained, think about it. And then they also looked into funding a number of studies to find an alternate bioretention soil media spec. And Curtis Hinman led a lot of this work, and he presented some of it today, and the knock-on work from it that's coming up now, but did a lot of studies over, I don't know how many years, Curtis, 10 years, 10 years, but a number of studies, and King County, Kitsap County, a lot of great partners, Seattle University, Western Washington University, University of Washington, all involved, making sure this stuff can grow plants and making sure it can not leach additional pollutants. We got to worry about that. We saw it with the compost. This is the media components we ended up with after testing, I don't know, 50 different ones, many different, you know, will they leach? Can you get water through at a high enough rate? All the different, and y'all have probably seen presentations on this very research. I'll breeze through it here, but these are the components up there. That is biochar, coconut coir, sand, and iron aggregate activated alumina. Those are our 5 now. This is just one data slide from that study, and what we're looking at here, I don't know, try to point with this virtual laser pointer, and then they can see it online. Is that working? Okay, cool. This is just looking at phosphorus. We looked at many other things, but the influent is here at about 160 micrograms per liter phosphorus. This is the compost, sand compost doing its thing, exporting. These are all effluent concentrations coming out of the bottom of columns, those pictures I showed earlier, so column study results. And lower the better here, because these are effluent concentrations. Number 4, that's amazing, right? Super low total phosphorus, and that's this very mixture I just showed the pictures of. Those components. This is some of the data that moved us towards getting this alternate bioretention soil media spec approved, the HPBSM. You can go to Ecology's website and get the spec on that. Meanwhile, in Bellingham, they have a big water body there that they care a lot about when it comes to phosphorus loading. And so, this new spec came out. Now you can build bioretention within a quarter mile of a sensitive receiving water because it doesn't export phosphorus. In fact, you saw it, it decreases phosphorus a lot. So super exciting. They can start building this stuff along the shores of Lake Whatcom. And that's what the county decided to do. That was the background. We're getting to the project. So this is the Geneva swale, and you see that on the right there, the basin for the Geneva swale, rather large, and we're treating across like 2 city blocks. It's not a city though; it's a really low density residential neighborhood. There's a nice photo of it. The problem was the existing facility there was failing. There was some nearby basement flooding. It was fouling because there was a lot of baseflow coming in to this system because it's a big pervious basin, so it's like a seasonal stream almost going along the road. And this was causing the media to foul on this existing system. And in fact, put a big pipe and just routed around the whole thing. So it was offline. And there was an opportunity here to retrofit this thing and improve it. So that's what we did. Those are those problems listed. And redesigned it as a regional facility using the HPBSM. You know, at this point, this is one of the first installations at scale of this new media in the field. It had been tested and proved in the lab. So we got an SFAP grant. And so again, thanking Ecology for that, to have this rebuilt. Lined it and put in the HPBSM, put in a dosing siphon with flow distribution. That was a pretty cool thing about this. Inherent problem with regional facilities is it's always baseflow coming in because you have these bad basins. And what do you do with that? If you try to put it through a filter, the filter's wet all the time and bad things happen. If you route it around, you're not treating any of that, right? And there's phosphorus in groundwater, and so they care about treating the groundwater. So our solution for that here was this dosing siphon, and we'll get into that. Design flow is half a CFS, 134 acre basin. This is what it looked like under construction. It was sort of a somewhat steep street. So we put a bunch of check dams in, Jersey barriers that we bolted some metal onto to flatten it out. So a whole bunch of tiers lined the whole thing. There's a cross section, the main media layer, and then the polishing layer underneath, layer cake. There's a picture of the dosing siphon going in. There's a big old holding tank with the siphon downstream of it. So there you have the big fat pipe on the side, and then the dosing siphon with a blow up of that down. This is used really commonly, apparently, in on-site septic, but not so much in stormwater. First stormwater application of this that I know about. And then a hydrodynamic separator upstream of that. And that was all upstream of the actual cells. So there's a cell filling up with water on the left. That's what it looks like. The water pulses into this thing and sort of fills up and breathes. And it goes, water goes in and down. And we monitored it full sort of tape style monitoring, automated samplers, weirs, pressure transducers, telemetry, all the intelligence baked in to get the data. So let's get into that. Oh, that's a lot of numbers all at once. I have some plots of this too, but really you can just sort of focus on the colors here. And we have 3 bins we're looking at here for comparison. All of them have effluent concentration, percent reduction, and the N value. So first set here is just the 60/40 bioretention. You see the N value is really high. This has just been compiled from studies all around the region. And so these numbers are pretty robust in terms of how typical 60/40 bioretention performs. You see it exports phosphorus, right? Big red bar there. And it exports a bit of copper. And we saw that in Amy's data too. So we see this pretty consistently now, right? But it's really good for zinc removal, good for fecal coliform, for TSS. The middle section of data is the column studies for the HPBSM. And they were really promising, of course. And then the final set of data is our actual results from the field. Our N values are on the end there. At this point, we're not quite done. I think we need one more event. 17, between 12 and 17 N value. Turns out we're doing really well and essentially equivalent, if not better, than what we saw in the lab. And you see there's fewer green bars over there, but that's all driven by lower influent concentrations. This was a really clean basin, and we had clean water coming in, and we were able to achieve that very difficult thing in stormwater treatment of getting cleaner. And so I have some box plots to show that. We're looking first at bacteria here, and this is just like a bacteria home run. This is a log scale, right? And so look at the effluent concentrations are almost all below 10 CFU per 100 milliliters, and you see our influent concentrations are much higher. The county cares a lot about bacteria loading to the lake as well. It's their drinking water supply. So bacteria and phosphorus were their 2 things that they cared about the most. 97% reduction is about as good as you can do. These were the metals results, and some of the most impressive to me, just because copper is so hard to remove. It's a tricky one in the stormwater world, and particularly low concentrations of copper. And so you see, it's not an impressive number when it's compared to the rest of the boxes in this grid. The one on the top right is dissolved copper. It's only a 26% reduction. but it's a 26% reduction when the influent concentration is 2, which is almost the detection limit. And so that was really, really cool to see. The water was just coming out crystal clear, and you can see it completely wiping zinc out. Those are the bottom rows, 87% and 74% reduction. Again, when the influent concentrations are low. So great metals performance. Nutrients. Total bottom left is the one that we mainly cared about, total phosphorus. And so the goal there, the TAPE goal is a 50% reduction, and your approved phosphorus treatment facility. We're getting 75% reduction, again, with the influent concentrations below the typical TAPE goals, where TAPE says, oh, that's too low, throw those data out. You don't have to treat that low. Well, we are that low, and we're still getting it 75% lower. And this is because, you know, the iron in there, the activate alumina, these things love to absorb phosphorus. Great for TSS, you know, less good for nitrate. I think the orthophos result there is just because it's almost detection limit at the inlet. It's just like same in, same out. Okay, those were those data. While that was going on, Curtis and Chelsea were doing their study in the lab, right? And seeing that these same media, the HPBSM, that's what we have in the field here, as well as looking at the 60/40 in these columns, we're doing really good at removing 6PPD-Q. Now we wanted to test that out in the field. So Chelsea said, hey, I heard you're doing that Whatcom County project. Can King County jump on and look at PFAS and 6PPD-Q as well, since you're collecting samples? And we said, sure, let's make it a party. And so we did that. Curtis, you already presented this slide, right? Yeah, I think so. So I don't need to rehash what he presented, but did really well. The HPBSM for removals, the influent concentrations were really high, and these are all the effluent concentrations from the columns. So we had a good idea that it would do well, the HPBSM in the field. I was just surprised to see how well it would. So I've been doing this for 20 years and probably monitored over 100 different stormwater treatment systems with a similar approach. And you don't see this. You don't see influent concentrations get reduced at every effluent concentration being non-detect when the detection limit is 1 nanogram per liter. And so that was really surprising. This thing wipes out 6PPD-Q. So that's really lucky. Happy to see those results. And I think Amy showed a similar slide with almost the same results, like the one she's doing in Spokane. It's almost all non-detect at the outlet. That's just really great to see that in 2 different locations. Good confirmation. Oh, just a reference line there. That's that 12 nanograms per liter. We're below that at the effluent, which is great. Now this is cool. We haven't done much PFAS in stormwater in this region yet. We're a little behind some other regions, and really a lot of the PFAS data and analysis has been done at contaminated sites. And so you see some performance data with really high influent concentrations, but that's not so applicable to what we see in stormwater around here. So I think Brandon Steeds, who was here earlier, is giving a presentation after this one on more PFAS in stormwater, but excited to see these data here. That's a lot, and the screen's tiny up here. I don't know how many PFAS species there are. I think it's like 20,000. It's a really high number. So we analyzed like 17 of them. The ones most commonly talked about are PFOS and PFOA. So I'm highlighting those down there. You can see, unlike the other numbers, we didn't do as well for PFAS. Influent concentrations, again, were really low at the inlet, so I didn't expect much of a reduction. But you know, it was sort of same in, same out, if not a little higher at the outlet than at the inlet. So some sort of minimal leaching of PFAS potentially going on from something in the system, maybe the liner, I don't know, but not at concentrations you would particularly care about. Like the MCL for drinking water is 4, and that's really low. I heard they might actually change that, and it's been in the news lately. So yeah, you see that line, that MCL line on our data, but when you look at the numeric water quality criteria for Washington, the chronic criteria, they're way off the chart, orders of magnitude higher. So we're talking about environmental concentrations that are not super relevant here, but still interesting to see PFAS and stormwater and treatment of it. Want to do more of this, maybe at a dirtier site. We need to do this at a dirtier site, maybe at the Ship Canal or something. That was all water quality. I haven't gotten to. Okay.
Oh, yeah.
Oh, yeah, so I told you this thing was pumped, right? It's really, thank you. That's a weird hydrograph. So this siphon is pulsing water through this thing because we want a chance to dry out between the pulses, right? So we're looking at, again, the axes are tiny, you can't see what's going on, but this is October to January. And the flow rates are getting up to a peak of like 65 GPM coming out the underdrain. We monitored one section of this long facility, not the whole thing. And so then we scaled the results up to get our total volume treated. And these are underdrain flows coming out the bottom. In the end, when we do scale that up, what we were able to treat was over the course of one water year. We've gone beyond one water year now, but just for water year 2024, 5.1 million gallons were treated in this thing. And of course, we have to convert to Olympic-sized swimming pools. We got 8 of them with this thing in one water year, with all that amazing treatment going on that we showed with the box plots. And a really fun looking hydrograph, which is hard to sample. Like poor Skylar, he was out there trying to grab samples, and the water would just disappear and you'd have to wait an hour for the next pulse to come. So here's what it looked like before and after. And some conclusions is that the thing cost $700,000 to rebuild. It removes about 8.5 pounds of total phosphorus per year by our calculations right now. Again, we haven't written the final report yet. This is early stuff. There's no more basement flooding. The 6PPD-Q is completely wiped out, which was great to see. And then it's meeting forested conditions for total phosphorus, which is really great because that's the objective in the region with the TMDL, is to get developed neighborhoods to sort of act like forested areas when it comes to phosphorus loading, and it's able to do that. So I just want to say thank you to all these partners. I didn't just cover one study there. I think I covered 8 studies, and so a lot of people were involved through the years. But for Whatcom County, Cody Swan and Craig Olison were leading this whole thing. Our Herrera team, our engineers, Chris and Neil and Colleen. Skylar, I already mentioned him. He was out there in the field all the time collecting these data, and Lacey and Nick and Sam. The labs Exact, and then ARI and King County Environmental Labs. They were all great to work with. And of course, Ecology funded almost all these studies I presented. So it would be a long list, but Morgan, Shelby, Tanya Williams, Chelsea, Doug, and of course, the King County folks, Janae and Chelsea, and finally, of course, Curtis. So thanks everyone who was involved in all these studies. And with that, I will take questions. This was a Chris Webb invention. He's done a lot of like on-site septic stuff and used to do this. There's just a big old pipe on its side here, and it slowly fills up with water because the siphon's closed. And then once it comes up, there's like a float in there, and it comes up and it breaks the siphon and it starts sucking the water down. And because it's a siphon, it can suck all the water down to the bottom. And so it like pulses all the water out, and then the siphon closes because the float comes back down. And then the siphon breaks, and then it fills back up, and it just sort of pulses like that.
Sucking all that baseflow.
Yeah.
To dry out.
Yeah. Like while the thing is filling up, the media downstream is drying out, because it takes a while to fill up, and then it hits it all out. Yeah. And it's nice because it doesn't need like a SCADA system to run it or anything. It's just passive. That's a good question. For some reason, that never happened. I think we have the upstream hydrodynamic separator, and it's got like a floatables baffle. I don't think we've ever gotten chunks in that big pipe because this siphon has never failed. I've been monitoring the hydrograph, but yeah, that would be a failure point. We did have upstream of this whole thing is a flow splitter, because otherwise the basin would be twice as big. And that flow splitter had an orifice that would clog, and so yeah. Yeah, they're similar. I think the only differences is that instead of a sand, it uses a gravel, the post system to get a much higher flow rate through it. But because you have a much higher flow rate, you have less of a contact time. And so to compensate for that, the activated alumina content is upped because so you want to store more stuff more quickly. So the post media has more activated alumina in it and a gravel. Otherwise, they're very similar. Good question. Not for this part of it, but there was implications downstream because the water comes out really fast. And so, these cells get sort of hit with water pretty fast. And so there was some erosion that was occurring. And so the county's been working on trying to figure out ways to dissipate the energy even more and prevent the, mulch floating is always an issue with bioretention. It's even more of an issue when you have a current, and so I think there's some learning there about how to do downstream design to make sure things don't get a little blown out. So they're putting down erosion control stuff now out there.